I think we should get started. Hello, everyone, and welcome back to the second part of the Plant Cell Atlas Symposium. I'll be chairing this session, which will focus on spatial profiling of plant tissues. We have four really excellent speakers lined up. The first is going to be Tatsuya Nobori, who is a HFIT fellow in Joe Ecker's lab at the Salk Institute. He's going to tell us about multiplex single cell three-dimensional gene expression analysis in plant tissues. So go ahead. Yeah.
Okay, hope you can see my slide and hear me okay. Let me pull out the pointer first. All right. So thanks, Ben, for the introduction. And I also thank all the organizers for putting this wonderful event together. I can imagine a lot of efforts has been put here. Today, I'd like to introduce PHYTOMap, which is a new method that enables multiplexed single cell and 3D spatial gene expression analysis that can work in thick plant tissue, such as whole mount plant tissue. So I hope you'll find PHYTOMap useful for your own projects, or at least you enjoy this Christmas tree. So before talking about spatial gene expression analysis, I'll start with a little bit about single cell gene expression analysis. So as you might know and heard already about in the morning session, single cell transcriptomics has been instrumental for many kinds of atlas generation. Here I'm showing a few examples from recent studies. There are non-plant studies. And single cell transcriptomics is powerful in classifying cells into populations based on their molecular features. And these populations can be distinct cell types or cell states, and the classification of them is an important first step towards the deeper understanding of the building blocks of the tissue. And more recently, single cell transcriptomics has been applied to many plant species and different tissues. And like many others, our lab has been putting a lot of efforts in generating different kinds of atlases of plants and also mammalian brains. And this is a teaser for a poster presentation tomorrow from my colleague, Travis Lee. So in this project, we have generated an Arabidopsis transcriptome atlas that covers the entire life cycle of the plant. So Travis and I have selected 10 distinct tissues from across different developmental stages, including germinating seeds, growing seedlings, rosettes, and stems, flowers, and sleeks, essentially closing the circle. We've profiled single nucleus RNA sequencing of over 0.8 million cells and identified hundreds of distinct cell populations, the clusters, based on their transcriptome identity. Oh, I see. Okay. And we've tried to annotate many of them with known cell types and states, but there are also a lot of clusters remain unannotated, which is really exciting. Maybe some new discoveries awaiting. And we've been systematically analyzing the transcriptomes of those hundreds of cell populations, trying to understand what defines cell types and states all across the life of the plant. So Travis will tell you more about it. So please go to poster number 16 tomorrow. And in such an atlas project, an important task for us is to annotate those cell populations and also validate de novo identified cluster marker genes, because these can be cell type marker genes or cell state marker genes, which could be used in the future studies. And this is where spatial gene expression analysis is important. So what the current plant biology community would do for this task is to generate transgenic report lines, so expressing GFP or other fluorescent proteins under the control of promoter region of your gene of your interest. And this works really robustly if your tissue is transformable, and it allows whole mount imaging using confocal or light sheet microscope. And also you can even do a live imaging, which is really fascinating. However, it also has some limitations, especially in the context of single cell omics studies. Well, first limitation is generation of transgenic lines is time consuming. It takes months, and it might be okay for a couple of transgenic lines, but if you have hundreds of clusters with many hundreds of candidate marker genes to validate, it's going to be a daunting task. And second, when you have more and more cells with better technologies, you will be able to resolve cell populations at really high granularity. And at this point, a single gene might not be sufficient to really define a cell state or a cell population. It might take multiple genes in combination to really define a single cell population. And in terms of such multiplexing of spatial gene expression analysis, this transgenic line approaches are limited. So you can go up to 3 to 4 genes if you work hard, but not more than that. And lastly, in such a heterologous expression system, it might lack the native genomic context, such as enhancers. So if you take a fixed length of the promoter regions, like 1 kb, 2 kb upstream of the gene, it may not faithfully recapitulate what the actual gene expression in situ. So because of these reasons, we need some methods for simultaneous spatial mapping of many RNAs without making transgenic lines. And there are exciting new technologies emerging that can do exactly this. And that is spatial transcriptomics. It comes with different flavors. I'm not going to review all different sorts of techniques, but there are array-based technologies or microscope-based technologies. They are all good at simultaneously mapping a large number of genes on the tissue. And we are also using microscope-based spatial transcriptomics. The assay is commercially available, and it's really, really great. However, it also has some limitations. The biggest one is the tissue sectioning. So all the commercially available spatial transcriptomics platform requires tissue sectioning, so typically very thin 10 micron, like a single cell layer sections is needed. And sectioning itself can be really tricky, especially for tiny plant organs such as Arabidopsis root tip, which is a very important model for many plant biologists. And also, if you can get a nice section, you're going to lose the rest of the tissue, right, which may contain important cell types. Or in my case, I'm interested in plant microbe interactions. So, so the sectioning could cause a loss of microbial colonization information. And if that happens, the interpretation of the data on the thin section can be quite difficult. So that's why we need some sort of spatial transcriptome kinds of method that can work in much thicker, ideally whole mount plant tissue. And that was the motivation behind the development of PHYTOMap. So again, PHYTOMap is a solution to that problem. And it builds upon many different technologies, primarily developed in a neuroscience field. So I'll briefly explain that. So it's a little, I don't know how you see the panel. It's something funny is going on, but I hope it's not too destructive. So the first step is to first you fix your tissue, the whole mount tissue, so you don't have to worry about gene expression changes in downstream processes. And then next important step is you replace all the targeted mRNAs with DNA probes. And each DNA probe has a target-specific barcode sequence, which will be later targeted by FISH. After hybridization, those DNA probes are amplified in situ for hundreds of times, and then cross-linked with the stereo matrix or between with each other so they don't move. So at this point, amplified DNA replaced the original mRNAs at the original positions. And then single molecule FISH is performed, targeting those target-specific barcode sequences. And please remember that the barcode sequences are now amplified hundreds of times, so they can incorporate hundreds of FISH probes, giving a really strong signal. And that's actually one advantage of PHYTOMap compared to traditional single molecule FISH, where a bunch of fluorescent probes are tiled directly onto RNA molecules. So this could be dozens of probes, but not hundreds. So PHYTOMap will give you much higher signal noise ratio, which is crucial for deep tissue imaging in such a highly autofluorescent tissue. And a bonus is that now all the information is encoded in DNA space, not the RNA space like SM fish. So it's much more stable, allowing multiple rounds of imaging that I'm going to tell you in a second. And also, this will give you a peace of mind, so you don't have to worry too much about RNases and the degradation of your target RNA at this point. So here I'm showing an example of the actual signal from PHYTOMap in a very small region. So it goes through the hundreds of Z planes. Each dot is a single molecule derived, mRNA derived. Different colors mean different genes. Okay. So after this confocal whole mount Z stacking, All right. So after the detection of 4 genes with confocal, you strip away all the fluorescent probes while still keeping the amplified DNA still in place. And then you reuse the same tissue and perform single molecule FISH again. Next, now targeting a different set of 4 genes, and do this step again and again until you detect all the mRNAs you wanted to detect. Here I'm showing the actual whole mount tissues of Arabidopsis root tip across different imaging rounds. I chose Arabidopsis root tip because it's a difficult tissue for spatial transcriptomics using commercial technology, commercial assays. Here I targeted some putative cell type marker genes, and you can already see some patterns here. This is zoom in with cell wall staining. I hope you can appreciate specific signal coming from specific layer of the root tip, indicating the specificity of PHYTOMap. So PHYTOMap can dramatically accelerate cluster annotation and marker gene validation in single cell RNA-seq analysis. So as a proof of concept, I took this publicly available root tip single cell RNA-seq data from Rachel Shehan. That's actually what she has presented in the previous session. And here, so with our PHYTOMap, what you would do for validating those predicted marker genes for each cluster will be you generate individual marker lines, transgenic lines for such marker gene detection analysis. And this is going to be highly, really time consuming. But PHYTOMap will allow you to grab, whatever dozens of genes or several genes per each cluster and do one-shot experiment, and then it's all done without making any transgenic lines. So here in this preprint, I demonstrated such use case, taking some previously unvalidated cluster markers, so they are cell type marker candidates, and then I did PHYTOMap to detect and actually detect those predicted marker genes from expected cell types and tissue regions. So just looking at individual PHYTOMap images will be highly informative and powerful. But PHYTOMap can do more than this. So here, let me again emphasize some of the key features of PHYTOMap. So first, you can take images using exactly the same tissue over the course of FISH. So at the end of all the experiment, you can combine all the images together into one tissue image. Also, it has a single RNA molecule resolution. You can basically count each spot to do sort of a semi-quantitative analysis. And of course, it has single cell resolution. So single cell analysis is also possible. So to leverage all these, I developed a computational pipeline to do a single cell and spatial gene expression analysis using PHYTOMap. So the pipeline includes the 3D image registrations, aligning all those images across different imaging rounds, and automatic detection of FISH signal, the spots, and then you decode spots based on in which imaging round and which microscope channel you saw the spot. You can basically name the spot. For the single cell analysis, I perform cell segmentation using the cell wall boundary staining information. And then now you can assign those decoded spots into individual cells, and you can count those spots for each cell, resulting in this kind of cell by gene matrix, which is a standard data type for single cell transcriptomics. But here, each cell has the XYZ coordinate in the actual 3D tissue. Okay, so as a proof of concept of all the pipelines, I targeted 28 predicted or known cell type marker genes in the root tip. So it's going to be 7 rounds of imaging. I profiled 5 root tips, which gave me over 250,000 spots or UMIs, if you are familiar with single cell omics. So these are unique molecules. And then I got over 3,600 cells after removing the cells with low count. Here is an example of this transcript expression after combining all the images across 7 rounds of imaging, which already showed some interesting cell type patterns here. Here I show the reproducibility of PHYTOMap experiments, taking 2 different routes and quantifying 28 gene expression and showing really high correlation. So the PHYTOMap works robustly in a reproducible way. This heat map shows the expression of 28 genes across many cells, and you can appreciate some cell population specific gene expression, as you would expect from a set of cell type marker genes. So also the data can be shown in this kind of UMAP, as people do for single cell analysis. So here I again clustered cells together, and I could annotate those clusters based on their expression patterns. And what's interesting is that here individual dot is a cell with XYZ coordinate in the tissue. So I can put them back into the actual tissue, keeping this cluster label the same. So this is the mapping of those clusters. And here, basically those clusters recapitulated actual cell types and the regions in the actual 3D root tissue. So this demonstrates that the whole pipeline and clustering really captured real biology. Okay, so here, let me summarize again the key features of PHYTOMap. So to my knowledge, this is the only method that allows multiplex spatial gene expression analysis that can work in whole mount plant tissue. The protocol is relatively straightforward. It takes 4 to 5 days. It's kind of similar to standard in situ hybridization. So I would say any molecular biology lab can do it if you have a confocal microscope. And it's very low cost, about $80 per experiment, although you will need a bit of investment to really buy all the probes. But if so the per experiment cost will be much, much lower than commercially available spatial omics, although, they do different kinds of things. And importantly, PHYTOMap is transgene free, meaning that it can be potentially used for any kind of plant species, including non-model species, although, it will take some optimization, which I'd be happy to discuss. So I hope, you're interested in applying PHYTOMap to your system. And again, I'd be happy to discuss future application of this technology with you. And of course, I, developed PHYTOMap to address interesting biological questions. So as an example, I'll finish with telling you a little bit about what I'm doing and what I want to do in the future at the high level. So as I briefly mentioned earlier, I'm interested in plant microbe interactions since my PhD studies. And plant microbe interactions can be highly heterogeneous because different plant cell types have different molecular networks that interprets the external environment, such as microbial colonization. And the microbiome itself is highly heterogeneous in their spatial colonization patterns or their activities, depending on where they're sitting, with whom. So the combinations of those variables can lead to a wide spectrum of possible plant responses, or plant responses that each plant can possibly take. And different responses can happen simultaneously in asynchronous manner in the same tissue, making the single cell responses of plant tissue really, really heterogeneous. So to really resolve such heterogeneity in plant microbe interactions, one approach would be that, so first using single cell transcriptomics or other omics, you could comprehensively classify plant cells into those populations based on their transcriptome patterns. But at this point, you have no idea about where those cells came from in the tissue. But at least you have the molecular features of each cluster, which will be the marker gene expression. And then you can spatially map those marker genes using PHYTOMap in the 3D native tissue context. And what's really exciting is that PHYTOMap can be combined with other imaging technologies, such as microbial imaging. So then you can really combine those information together and really directly link microbial colonization and host gene expression at the single cell and spatial resolution. And I think this will be really exciting for future discoveries. And again, I'm really happy to discuss what else could be done using those technologies, probably in the community discussion session. All right, so with this, I want to thank to my PI, Joe Ecker, for a lot of support and freedom. And I also thank our colleague at Salk, the lab of John Corey, Shirley Wu, for letting me use their fantastic confocal microscope, which was really important for this project. I also thank our collaborators, Ryan Lister and Marina Oliva, for their help. And I also thank the funding, and thank you so much for your attention, and happy to take any questions.
Wow, thank you so much. That was an awesome presentation. There are a couple of questions in the chat. First from Pengbo Liang. Do you have a hard time regarding probe penetration, and can you, I guess, image root hair specific expression?
Yeah, so the probe penetration, so it was okay for the Arabidopsis root tip, which I believe is relatively soft tissue, but I can see problems with more mature area. In that case, you will need extensive cell wall degradation, which is actually a popular discussion topic in the morning session about protoplastin, but we can use similar protoplastin enzymes to overcome this. And the second question was the root hairs. So I was using the root tip of the Arabidopsis, so I didn't have root hair really, but I think in principle it's possible to see root hair gene expression using PHYTOMap.
Awesome. Another question from Ken Birnbaum. Is there a maximum number of probes that you can bind in succession, and when does a signal start to fade?
Yeah, that's a great question. So as far as I've tested, the signal intensity remained almost unchanged, at least until about 7-8 rounds of imaging. But also it depends on which channel you use. So if you use, I saw that the longer wavelength indicates earlier than the others. But at least 8 rounds are pretty robust. But I need to do it more, more systematic analysis.
Cool. Another question, how quantitative is PHYTOMap? Do you have issues with detection limit, or can you get the differences between small and larger cells? Do you need to normalize expression?
Sorry, it was breaking up. So was it the question from anonymous attendee?
Yes,
about the
yes.
the quantitative. Okay, how quantitative is it? Okay, yeah, that's a great question. So I've been comparing PHYTOMap quantification with single cell expression data, and I did see good correlation for many genes, but sometimes there are some off correlation, which could be because of the bad design of probes, or it could be, I don't know, maybe single cell data wasn't accurate. It's kind of difficult to conclude. But in general, yeah, the data correlates pretty well. So, but I would say semi-quantitative. So I'll need more improvement to make it really quantitative.
Great. Another question from Sam Debach. Do you need to optimize tissue fixation and clearing before full map?
Right. So the tissue fixation, I use standard method for in situ hybridization. I use what's called FAA. It was formaldehyde and acetic acid and alcohol, which worked pretty well. But maybe for other tissue types, maybe it's a good idea to look for protocols that are already used for in situ hybridization, so you can follow that. The same for clearing. I use Clear C, which I think many people are using, and it worked perfectly.
For roots, I mean, I guess Arabidopsis roots you get maybe a couple of cell layers without clearing. Is that the case, or is clearing absolutely necessary for this protocol?
Yeah, for the root tip, it's not necessary to clear it. Also, because PHYTOMap signal is so strong, so it also helped. But it doesn't hurt, so I will always use clearing solution as well.
Cool. More questions coming in. I guess a lot of questions about what tissues can be probed. Like, what are the main main limitations for like selecting a tissue that might work with it? Like what things to consider. I mean, you mentioned maturity, but are there other considerations?
Yeah, sure. So yeah, we are working on testing PHYTOMap in different tissues with collaborating with many different labs, and so we need to see the results. But so I've tried Arabidopsis leaves, for example, which worked okay. So the autophoresis isn't that a big problem because PHYTOMap signal is pretty strong. And I think the major bottleneck is the permeability of the tissue, as somebody asked already. And so the PHYTOMap requires the permeability, requires all the DNA probes and enzymes to get into the tissue. So the cell wall digestion step will be crucial for the wide application of PHYTOMap. But if I add another thing, it's a fixed tissue-based protocol. So unlike protoplasting, you can do really crazy thing. You can, throw a ton of enzymes and proteases to make holes on the cell wall.
Great. Question about the cost. How is that calculated?
Yeah, so I just summed up all the probe costs and divided this by how many experiments those probes will allow me to do. So that's why I said you have to pay more than $80 because the probes, they don't sell probes in a really tiny quantity, but it's not. So I think practically it might cost about $1,000 or $1,500 to $2,000 to at minimum, and then you can do about 10 or 20 experiments, which is not too bad, I think.
And then last question in Q&A from what is the subcellular resolution? How finely can you resolve something?
Yeah, so in theory, it should be subcellular. The resolution is subcellular, but practically, depending on how fixation worked well, so the molecules may move, or the vacuole collapsed during fixation, etc. So this may compromise the subcellular resolution. But at least at this point, we do see nuclei and also the cytosolic sort of distribution of transcripts in some cases. But sometimes, sometimes a cell is filled with spots, which may be due to the collapsed vacuole. But I still believe that still that the cell contained the original RNA within it. So there might not be much diffusion to the neighboring cells based on the marker gene expression analysis.
Awesome. I had one maybe last question.
Sure.
So did you design the probes yourself?
Yeah.
And what kind of considerations goes into probe design?
Yeah, so it's similar to like when you design PCR probes, it should be specific. But there are some special consideration because I need a pair of probes, like a tandem probes sitting next to each other on the mRNA. So that you need to design two specific probes next to each other, which will limit the possible probes. But in many cases, I can find such regions in a transcript.
Great. Well, thank you. Thank you again for a really fantastic presentation. I think there's a lot of interest in your method. So you'll hopefully get a lot of follow-up emails.